Good afternoon, everyone. Dan Mikol, Chief Medical Officer at NervGen. I think we're probably the smallest company this afternoon. We have about 12 or 13 employees. So as an overview, in my presentation, I'm going to first begin by reviewing the scientific background behind NVG-291, summarize results from preclinical efficacy studies, and then I'm going to describe the approach that we're taking as we hope to successfully translate the positive results from animal models of spinal cord injury to our proof of concept trial. And in the proof of concept trial, we're going to include both clinical outcome measures as well as electrophysiological outcome measures to measure functional recovery after treatment. Because electrophysiological measures are not routinely used in clinical trials, I'm going to explain the rationale for why we're including them as surrogate efficacy measures in order to increase the probability of detecting an efficacy signal. I'll summarize our phase 1 results, and then I'll end by reviewing the study design for our proof of concept trial that will begin in the next couple of months. Okay, so let's talk about the scientific background. Since in 1990, chondroitin sulfate proteoglycans, or CSPGs, were first demonstrated to be substantially upregulated in and around the glial scar after spinal cord injury and to inhibit neural repair mechanisms. And in 2009, protein tyrosine phosphatase sigma, PTP sigma, was identified as a cellular receptor for CSPGs that could mediate these inhibitory effects on repair. In 2015, Jerry Silver's lab at Case Western Reserve developed a peptide therapeutic targeting rat PTP sigma, which they termed intracellular sigma peptide, or ISP. ISP has two parts. It has an amino terminal TAT tag that enables translocation across cell membranes and the blood-brain barrier. That's shown in red. It also has sequence derived from the intracellular wedge domain of rat PTP sigma, shown in purple. NVG-291 is the human analog of ISP. It has exactly the same structure with the exception of a single amino acid difference in the wedge domain sequence. And since it was first developed in 2015, ISP has been tested in a number of different preclinical models of spinal cord injury and has been shown to improve functional recovery by enhancing repair mechanisms such as axonal regeneration, plasticity, and remyelination. So I'll show you some of these results. This slide shows results from acute and chronic studies. On the left is an acute thoracic injury model where ISP treatment was initiated just 1 day after the injury. And on the right is a chronic cervical injury model where treatment was begun 3 months after injury. In both cases, the treatment with ISP was administered as a daily subcutaneous injection for either 7 weeks on the left or 8.5 weeks on the right. And both of these studies show significant motor recovery, but the outcome measures that are shown on the slide are different. On the left, the BBB scale is a measure of mobility, and on the right, the four-limb locomotor rating scale is a measure of forearm and fine digit function. So we can't compare the trajectory of the curves, but both of these studies show significant motor improvement. And it's worth noting that results like this for ISP have been replicated many times over by several different investigators. And in these studies, the focus has always been on assessment of motor and locomotor function. 2 of the studies also looked at bladder function and were able to demonstrate improvement in bladder function. So I'll show you results from one of those studies on this slide. And you can see that ISP had a nice dose-dependent effect, improving bladder function after spinal cord injury. So as we began the transition from these positive results in animal models of spinal cord injury to planning the investigation of NVG-291 in humans with spinal cord injury, we had 2 main hypotheses. First, that NVG-291 treatment will improve neuronal connectivity across the level of the injury, and that we can measure this improved connectivity electrophysiologically. And secondly, that NVG-291 may be effective in individuals with subacute or chronic spinal cord injury, or both, in view of the preclinical efficacy results. The question was, how do we optimize the probability of success as we enter our first proof of concept study? And the answer, I think, lies in careful clinical translation. So what we have learned from preclinical studies is that ISP has demonstrated efficacy in both acute and chronic intervention models. In our clinical trial, we will actually enroll 2 separate cohorts, individuals with subacute injury and chronic injury. The preclinical models are motor incomplete models. Our clinical trial will enroll individuals who are motor incomplete. That is, they have some residual motor function below the level of the injury. Preclinical models have looked at both cervical and thoracic injury with ISP. In our clinical trial, we're going to focus on cervical injury, really because the chronic studies with ISP were cervical injury models. ISP was always administered as a daily subcutaneous injection, and so moving to humans, NVG-291 will be administered as a daily subcutaneous injection. In animal studies, a very broad range of doses was tested. Doses as low as 0.007 milligram per kilogram to 0.32 milligram per kilogram, and this entire dose range did show efficacy. So as we move to the clinical trial, we have selected a fixed dose for our proof of concept trial that will result in exposures that are at least comparable to, if not higher than, exposures in animal at the highest efficacious dose tested. The treatment period in the preclinical studies was 7 to 8.5 weeks. Our clinical trial is 12 weeks. And again, the preclinical studies have focused on assessing motor and locomotor function. In our clinical trial, our clinical measures will also focus on motor and locomotor function. But in addition, we have included electrophysiological assessments as a surrogate efficacy measure to monitor descending connectivity. So let's shift gears and talk a bit more about surrogate efficacy measures. Robert Temple, who used to be at the FDA, defined it as an outcome measure used as a substitute for a clinically meaningful endpoint where changes induced by a therapy on a surrogate endpoint are expected to reflect changes in the clinical endpoint. The advantages of a surrogate measure are that it's more sensitive to change than clinical measures, and this can enable shorter studies and smaller studies. A couple of quick examples of surrogate measures outside of spinal cord injury. First, in cancer. In clinical trials of cancer, the clinical endpoint of interest is often survival. The surrogate endpoint that's often used in proof of concept studies for many types of cancer is reduction in tumor size. And for HIV, the clinical endpoint of interest is really time to AIDS diagnosis or death. The surrogate endpoint that has been successfully used in HIV trials is reduction in HIV viral load. Okay, so now we come to spinal cord injury. What we would like in a surrogate measure for spinal cord injury, first of all, because spontaneous recovery after injury is variable, we'd like a surrogate measure to predict clinical outcome during spontaneous recovery. But for it to be useful in clinical trials, we'd also like changes in a surrogate measure to be correlated with changes in clinical endpoints after intervention with a putative neuropreparative therapy. Well, electrophysiology would appear to be a good surrogate efficacy measure because what we're really measuring is the integrity of electrical connectivity, the wiring between the brain, the spinal cord, and the rest of the body. Let me talk about motor evoked potential specifically, or MEPs. First, an MEP is elicited using a magnetic coil placed on the scalp over the motor cortex, and the neurons in the motor cortex can be activated and send an electrical signal down through the spine and eventually to the target muscle of interest. And with a recording electrode on the muscle, you can detect an amplitude, the strength or magnitude of the electrical signal reaching the muscle from the brain. And there's also a latency, which reflects the speed of electrical transmission, how long it takes to get from brain to muscle. So these are useful things that can be measured. Of course, after spinal cord injury, the interruptions can be disconnected, or the connections can be interrupted or lost. So what may happen if you're measuring MEPs is that the amplitude may be reduced or it may not be measurable for a given MEP, and similarly the latency may be prolonged or undetectable. So motor evoked potentials can tell us a lot about motor connectivity after an injury, and these can be followed over time non-invasively. So over the next 3 slides, I will address the following 3 questions. First, does preclinical evidence support an association between changes in motor evoked potentials and functional recovery? Is there such evidence in humans? And to what extent have electrophysiological measures been used in clinical trials? So the literature does very much support the prognostic value of MEPs in preclinical models of spinal cord injury, and a nice example is shown on this slide. This is a rat cervical contusion model. There were 2 different severities of injury, as you can see on the left. And this study just followed spontaneous recovery of the animals. And in the middle panel, we're looking at recovery of motor evoked potential amplitudes. And you can see that right after the injury, the amplitudes are very much depressed, more so with the more severe injury, and that they recover over time, and recovery is better for the less severe injury. This study also showed very strong correlation between both MEP latency and amplitude and functional recovery. Now in humans, motor evoked potentials obtained around the time of the injury have been shown to predict neurologic recovery. And during spontaneous recovery after injury, and by that I mean over the year or so after injury, looking at spontaneous recovery, it has been shown that there's this very strong correlation between MEP amplitudes and the extent of neurologic recovery. And a lot of this evidence comes from the European EMSCI database. And I found it interesting, a paper published 20 years ago, this is reference number 5. This was published by Armin Kurt and colleagues who work very heavily in this database. They suggested that quantitative changes in motor evoked potentials might be used to assess investigational therapies directed at spinal cord repair because they're more sensitive than clinical measures. And that was 20 years ago. Now finally, there's also some emerging evidence from electrical stimulation studies that the greater the improvement in an MEP amplitude, the greater the clinical improvement. A few weeks ago, I went on to clinicaltrials.gov. I was curious to see to what extent electrophysiological measures have been used in clinical trials of spinal cord injury. This is what's based on information that's posted. And I found that there were 6 relevant industry trials ever conducted that used evoked potentials, and these were all in motor complete populations. There were 101 non-industry trials ever posted, and these were mostly in motor incomplete populations. And one of the examples below is the study with anti-no-go at the University of Zurich. This is just a glimpse into clinical trials that are using evoked potentials, but I think the glimpse may be telling us that industry is missing an opportunity to use electrophysiological measurements as surrogate efficacy measures in proof of concept trials. Okay, so the last part of my talk is on NVG-291 clinical development. So we have conducted a phase 1 trial of NVG-291. Subjects were randomized to receive either a single dose of placebo or NVG-291 at 6 different dose levels, or multiple doses over 14 consecutive days at 4 different dose levels of NVG-291. And what's important to take away from the dose levels we tested in this trial is that the doses exceed the very broad range of doses tested in preclinical efficacy models and demonstrated to be efficacious. In terms of safety, the most common adverse event in the phase 1 trial was injection site related. These were more common in the NVG-291 group compared to the placebo group. The vast majority were mild, and there was otherwise no adverse event increased in the active group compared to placebo, and there were no effects on all of the other study parameters. So in our proof of concept trial, the overarching objective is to evaluate the treatment effect of NVG-291 on motor function. And in view of the preclinical efficacy results, we will pay particular attention to mobility, ambulation, and hand function. Our proof of concept trial is a single center trial. It's being conducted at Shirley Ryan Ability Lab in Chicago. The rationale for a single center trial is that this allows for uniform assessments and uniform rehabilitation, which can together help reduce variability of the results. And reducing variability is really important with electrophysiological measurements, which otherwise could be extremely noisy. So because this is a single center trial, we can use the same small number of assessors who have the same training and are using exactly the same equipment, technique, and analysis for all subjects in the trial at all study visits. Could this be possible with a multi-center trial? Perhaps, but it would require a lot of work and standardization. That's overcome by conducting this as a single center study. And it's feasible to do the study as a single center study because the cohort size is not large. We'll be enrolling 2 separate cohorts of about 20 subjects each. The treatment period is 12 weeks and the total study duration just 16 weeks. So the 2 cohorts we'll be enrolling are chronic and subacute motor incomplete cervical spinal cord injury, where chronic is defined as 1 to 10 years post-injury and subacute 10 to 49 days post-injury. Again, we're enrolling a motor incomplete population because the animal models are motor incomplete models. Some of the key eligibility criteria are shown on the slide. Of note, we are not using the Asian Impairment Scale to define the eligible population because it doesn't really help us. We are defining the minimum and maximum level of motor function in these motor incomplete cohorts based on ability to ambulate. Subjects have to be able to at least initiate one step, and based on grasp prehension scores. We're also requiring that motor evoked potentials be present in 2 qualifying muscles, the tibialis anterior, which is important for foot clearance during walking, and first dorsal interosseous, which is important for finger control and grasping. And you'll see why we have this requirement on the next slide. So in the study, we'll be assessing 10 muscle groups longitudinally with electrophysiological measurements as shown in the box at the top right. And these include the 2 qualifying muscle groups that I mentioned. Our primary objective is to evaluate the effect of treatment on corticospinal connectivity to these 2 muscle groups. The co-primary endpoints are the change in MEP amplitudes to the qualifying tibialis anterior and first dorsal interosseous muscle groups. So that's why it's important to be able to measure the amplitude at baseline, because our co-primary endpoints are based on the change from baseline. Now, assuming a treatment effect and variability of motor evoked potentials that's comparable to what has been seen with electrical stimulation studies with just 8 subjects per arm, this study has at least 80% power to detect a treatment difference favoring NVG-291. It's a much smaller sample size than what would be achievable with a clinical outcome measure. Here are our secondary clinical endpoints. There are 6 of them. The first 4 are actually performance measures: change in 10-meter walk test time, 9-hole peg test time, pinch dynamometry force, and grasp scores. So 2 through 4 are performance measures of the upper extremities. And then lastly, lower and upper extremity motor scores. We have additional secondary objectives related to other electrophysiological measurements, which are detailed on the slide. I won't go through them. And we have exploratory objectives that look at some other clinical measures like spasticity and advanced MRI imaging and plasma biomarkers. So in closing, ISP, which is the rodent version of NVG-291, has been shown to promote functional recovery in preclinical models of both acute and chronic spinal cord injury. NVG-291 has been shown to be well tolerated in phase 1, and now as we move to our proof of concept trial, we will be enrolling 2 cohorts of chronic and subacute motor incomplete cervical spinal cord injury cohorts. And in this study, we have a comprehensive panel of both clinical and electrophysiological measures to assess functional improvements. And again, the advantages of including the electrophysiological assessments, it's a lot of work, it requires a lot of expertise, but the advantages are this is a very sensitive, objective, and quantitative way of monitoring improvements in motor function. And it really maximizes our probability of observing as well as characterizing an efficacy signal in this proof of concept trial with NVG-291. Thank you.